right, you guys. Hello, everybody. So, as Linda already said, I'm one of the co-principal investigators on RID-HIV, which I looked it up, does stand for Reversing Immune Dysfunction. That's one of the Martin Delaney collaboratories, and it's good to see Jeff Taylor here on the call as well. He's our community liaison in RID-HIV. I'm also part of another collaboratory, that's the I4C collaboratory. That's the one Dan Baruch and John Mellors heading as well. So I love all these collaboratories, and it's great to be part of this one today. All right, so today I'm going to be talking about how we might engineer B cells for an HIV cure. And don't worry, for those of you who've never thought about B cells before, I'm going to talk a little bit about what they do. Okay, so I'm going to start with my summary for the community. Excuse me, and I'll pull that back up at the end to make sure that it still makes sense to you. So the key question I'm asking is, can we reprogram B cells to make custom antibodies? Whoops. So that, let me just see if I can get my little laser pointer pen. There you go. Key question is, can we reprogram B cells to make custom antibodies that could neutralize or block many different strains of HIV? Our key findings are, yes, we can, at least at this stage in the lab, and now we have to see if this can also work in animal models such as rhesus macaques. Why is this important? Because I think it could be a way to provide lifelong production in the body of antibodies that block HIV, and which unfortunately are difficult and not currently made by vaccination, which would be optimal. How is this related to a cure? Well, antibodies and their ability to neutralize or block HIV are an important part of how our immune system suppresses HIV and indeed all viruses. And you probably know a lot about this just because of COVID. It's taught everybody about the importance of antibodies. And finally, why should we be excited about this? Well, I think it's a completely new idea, not by me uniquely. There's certainly a group of labs that are excited about reprogramming B cells. We've not done this before. And if we can show proof of principle with HIV, I kind of feel like this is going to have advantages for other diseases, especially cancer and some inflammatory diseases, like, for example, inflammatory bowel disease. Okay, let's get going. Mm-hmm. So, first of all, just to think about what we mean by an HIV cure, and I think this is probably a fairly accepted definition right now, that an HIV cure could either be, the absolute gold standard, the complete removal of all HIV from a person. But if we can't do that, what would be pretty good would be to get to a state where somebody who has HIV can completely control that HIV without needing to take antiretroviral drugs and with no possibility of transmission. And I would also add maybe no additional consequences for their health as well. So this is kind of, what I think of as an HIV cure. And I'm sort of working in this space where I'm trying to think about ways we could hack the immune system to make somebody better at doing this control of HIV. Now, why don't we have a cure for HIV? And of course, there's 100 different answers, and each one of you can chip in on this. But one of the ways I think about it is how do we normally get cured of viral infections? So, COVID, measles, anything like that. So two things happen. First of all, our immune system fights the virus. The virus wins for the first days or weeks, and then eventually we can sort of take over. Our immune system takes over and fights the virus. And then in addition, if they are available, we can take drugs that also stop the virus from replicating. And in many cases, we rely on a combination of the two. And so that sounds great. But what's sort of puzzling is, these things also happen with HIV. Our immune system mounts an attack against the virus, and we have very good drugs that stop it from replicating. So why do we not get cured of HIV? So again, these first few slides as introduction are really to kind of just remind people of stuff they probably know really well. But one of the things we know really well is that when people take antiretroviral drugs or ART, although they can reduce the amount of virus in the body, that really leads to often undetectable levels of the virus. We know it's not gone away, and stopping ART can result in a rebound in most, but not all people. And so what we think of when we are, oops, why is that for, hold on a second, there we go. I think it's all these, I'm getting all these, waiting room, admit things.
Yeah, don't worry about those.
It's interfering with my little cursor. All right. So what we think of when we think about some sort of cure, treatment, or intervention, ideally, it would be a one-time treatment. Maybe you would take it while you were taking antiretroviral therapy, but then if you stop the ART, what would happen is either, the best case scenario, the HIV is completely removed, or the HIV stays controlled and stays undetectable, even though people aren't taking antiretroviral therapy. Okay, so, I hate this virus. I've been working on HIV since 1985, something like that, and I think of it as having these sneaky tricks that make it a challenge for our immune system to fight it, and make it a challenge for us to come up with ways to make a cure. So that's why we're here in 2023 with hopefully some good and sophisticated new ideas. But HIV is a formidable enemy, and I do think of it as having these sneaky tricks that some of which are quite unique to HIV. Okay, so the first one that I think you're all familiar with is HIV is a type of virus called a retrovirus, and as a result, it inserts its genetic material into the DNA of every cell that it infects, and it then becomes a permanent part of any infected cell. So, showing you what that looks like in a cartoon. Here we go. Here I've got HIV in orange, infecting a cell in green, and this is in pink. This is just to symbolize it inserting a DNA copy of its genetic material into our nucleus amongst our DNA. And then this inserted DNA then becomes the template to make new copies of the virus. So an actively infected cell is churning out new viruses. It's full of little pieces of HIV. And as a result, it's pretty visible to our immune system, which recognizes that something weird and foreign is going on. But one of the other big problems with HIV is that, as well as, if you like, showing its wares when it's actively infected, sometimes cells can be what we call latently infected. And this means that although the HIV DNA is inserted in that cell, the virus is, at least temporarily, switched off. It's gone to sleep. And as a result, because it's not making any HIV, it's completely invisible to the immune system. So these latently infected cells, they're like kind of stealth HIV infections. They can persist for a very long time, and the immune system doesn't even know that it should be trying to attack them. And that would all be fine if they stayed asleep. But we also know that they don't, that sometimes just randomly, or sometimes as a response to specific stimuli or triggers, some of these latently infected cells can get reactivated. And if they get reactivated, they then turn into actively infected cells. And so this problem of having latently infected cells that can be reactivated is why antiretroviral therapies are not curative. And I'll show you that on the next couple of slides. First of all, just a reminder, and I stole this slide from the curriculum slides, that latently infected cells can be in many different places throughout the body, including in lymph nodes, the blood, places in our body that have a lot of immune cells, such as the gut, and even in the brain. And so it really is a Herculean task when you think about how we could seek out and neutralize these latently infected cells, especially when they are so stealthy. And then this is why this ability of a latently infected cell to become reactivated, even randomly, is the reason that most people will rebound if they stop taking antiretroviral therapy. So as I'm showing here, we've now got a latently infected cell that's becoming productively infected. And so the reason we can't cure somebody of HIV, no matter how long they stay on ART, is because antiretroviral therapy does not get rid of these latently infected cells, and neither does our immune system. Okay, so that's HIV's probably, sneaky trick number one, if you like, which is a real big one. about why it's a persistent lifelong infection that our current therapies can't clear. Okay, so switching to sneaky trick number 2. HIV constantly changes to avoid our immune system, our immune responses. And before I kind of go into that, I'll just remind everybody that, again, if nothing, the last couple of years of COVID have turned everybody into a virologist. So I think everybody understands the idea that some viruses will mutate and change, and just as COVID is throwing up these new variants which contain new mutations. And it's not, if you like, something the virus does on purpose, it's something that happens kind of randomly. But as a result, there is always the opportunity for new variants or strains of viruses to emerge that are just a little bit different, just different enough that any immunity we've developed no longer works against them. Okay, so well, I've been talking about our immune response and our immune system. And one of the things I want to dive into is, what do I mean by that? So you guys have probably heard of things like T cells, maybe you've heard of B cells as well. There's lots of different components of an immune system, and I'm going to ignore everything today apart from the B cell, because this is kind of where I'm focused. So I would say B cells are a really critical part of our immune system, and this is obviously what my lab is working on. So here's a B cell. And B cells, what they do, their day job is they make antibodies. And every B cell in the body, if you like, makes a different antibody. They produce the vast, diverse repertoire of antibodies we make. And we need to do that because if you get infected with a new foreign, pathogen or a virus, you need to kind of go into this incredibly diverse repertoire and find the few B cells that happen to be making an antibody that is a really good fit for that pathogen. So I'm sort of showing this here. Here's an antibody, not to scale, specifically binding to HIV. And so we call antibodies that recognize HIV as anti-HIV antibodies, and a subset of those antibodies we say are neutralizing antibodies. So what we mean by that is you can imagine an antibody could recognize HIV and be kind of, if you like, stuck on the side of the virus and not interfering with the ability of the virus to still attach to and infect a CD4 T cell. So such an antibody is kind of neither use nor ornament, really. It's kind of sticking to the virus, but it's not stopping it infecting. In contrast, neutralizing antibodies are those antibodies that hit the right part of the virus, and they kind of block HIV from interacting with its CD4 receptor and entering into cells. And in a couple of slides, we're also going to be talking about broadly neutralizing antibodies that you've probably heard about, which do this trick even better. They can neutralize HIV, but they can neutralize multiple different variants and mutants and strains of HIV because they are finding and binding to something that's very conserved across the great diversity of HIV we have in people and across the planet. All right, so just to finish, these are antibodies. Ideally, they will neutralize HIV, and they're made by one type of our white blood cells in our immune system called B cells. Okay, so the problem is that HIV is one of the most mutating things on the planet. It's constantly changing to avoid our immune responses. And so what that looks like in practice is if somebody is infected with HIV, I'm going to call it the purple strain, their B cells respond and they start to make antibodies that recognize purple HIV. And so then the virus, because it's constantly mutating, goes, aha, okay, well, forget purple. I'm going to throw orange HIV at you now. And so the purple antibodies no longer work. And so then the body goes, it's kind of like a chess game. Okay, well, now I'm going to make orange antibodies that will recognize this latest mutant. And again, the virus continues mutating. And so now it has blue HIV. So this is this constant chess match between the virus and our B cells and our antibodies that means that we are constantly the virus is constantly one step ahead of the immune system, if you like. Okay, so coming to what my lab is doing, our goal is, can we engineer B cells to make custom antibodies, better antibodies that can outsmart this constantly changing virus? Okay, so if we were thinking about what type of antibody we'd like a B cell to make, if we had an ability to kind of, program them with any antibody we want, then the type of antibody we would want to put in would be one of these broadly neutralizing antibodies that has an ability to recognize lots of different strains of HIV and be somewhat, although not completely, resistant to the virus's efforts to mutate around it. So what do I mean by, actually, before I do that, no, let me do this first, yeah. So I'll come back to that slide, don't worry. So this is just going over what I've already introduced, the concept of broadly neutralizing antibodies, or BNAbs. So if you see this shorthand in my talk, that's what that means. So over here I have a cartoon of the envelope protein, or the spike protein of HIV, and the GP120 subunits. And this is just showing that we've now identified several different classes of BNAbs. And what's interesting is they don't just stick anywhere on HIV, they stick in certain very discrete locations, reflecting the fact that these, like this green part here, this yellow part here, which is the part of the virus that binds to CD4, so we call it the CD4 binding site. And this reflects the fact that although I said HIV is constantly mutating and can change, there are going to be certain parts of the virus that it really can't change. It can't change easily without losing function. So you can imagine that the part of GP120 that specifically docks onto the CD4 receptor, that's got to maintain its shape and structure to maintain its function. If it started mutating this, it would no longer be able to bind to CD4. So that's why, for example, any antibody that manages to pull off the trick of recognizing this yellow part of GP120 is a pretty good antibody, because it's recognizing something that's not going to mutate much. Okay, so that's the concept of broadly neutralizing antibodies. So they are special, and they're pretty rare antibodies that can recognize many different varieties of HIV and therefore are somewhat resistant to viral mutation. You can make recombinant or artificial antibodies. You may have heard of monoclonal antibodies, for example. Again, I keep coming back to COVID. It's a great teaching example. But, people have been given monoclonal antibodies that recognize the COVID spike protein. So in the same way, injecting people with recombinant or monoclonal BNAbs has shown promise in clinical trials and in animal studies, especially when more than one BNAb is given at a time. You can imagine, giving somebody, say yellow and green and blue antibodies at the same time puts a massive restriction on the ability of HIV to kind of mutate against all of these weapons. And so having combination antibodies in the same way that we have combination antiretroviral drugs is important to do. And again, I've already mentioned this, but let's just stress it, that ideally we would want a vaccine that can make people make their own BNAbs. But unfortunately, most of the time when people are infected with HIV or vaccinated against HIV, the antibodies they make don't target these BNAb target sites, and they kind of go for, if you like, the rest of the bling on the outside of the envelope protein. There's been some progress recently, and there are certainly ideas about how we could make vaccines that could create BNAbs, but at the moment we're not there. I'm going to go back a slide. I skipped over this one just because this is just to kind of remind everybody that, antibody therapies are already an incredibly important part of medicine. People get injected with monoclonal antibodies or recombinant antibodies, and they can be very important and powerful in both preventing infections as well as treating infections for where we currently don't have a vaccine or a drug. But because they make antibodies that, will eventually disappear, if it's a chronic infection or chronic disease, you need to do constant re-injections, and there can be really high costs involved. Over here, I pulled up data. This was from 2016, so God knows what the prices are now. But this is just showing you the annual cost of some of the FDA-approved monoclonal antibodies. And you'll see, especially in the cancer space, some of these are eye-popping, okay? But even for some of the ones that aren't against cancer, people who have conditions such as inflammatory bowel disease get, I don't know if it's monthly or quarterly infusions of antibodies to help them maintain health. So there are a lot of antibodies that are given multiple times over a number of years. And so even if the price is only, $50K a year, it's still going to mount up, and it's also not pleasant to have to go and get an infusion of antibodies. So they're awesome. I think we can do better.
So can I ask you a question, Paula? I mean, it's about doing better with this. I mean, I remember the first meeting that Dates had about this, and there were a lot of industry people there talking about how everything was so much more streamlined than it used to be, and how it was going to cost less. And, and in other words, you just can't believe anything that they say. I've talked to Carl Diefenbach at Dates about this at length, and he, assured me, there'll be agreements and all these sorts of things. And, I've been working with drug companies or manufacturers for 30 years, and they, unless there's laws that they have to comply with or stop them from these exorbitant prices, they're not going to stop. So that being said, if it was a home run, I could see that. But it's gone from one dose, one antibody to now we need frequently, re-injections and all of the expense that comes with that. And then we need, it needs to be combination therapy. So, it's kind of crazy when you have people that have one pill once a day. I think the hope was, as you say, that it would have a vaccine reaction. I mean, that was what the last time that I asked this question, it was like, okay, well, we hope that they'll continue to keep manufacturing and carrying on in your immune system, but we don't see that. So it is what you're saying, to make a long story short, that we should keep going with this because we have seen some progress, and that maybe we will find that, that sweet spot where find something that acts like a vaccine or what? I don't know. I'm kind of.
So gosh, you've touched on so many things there, Linda. So I think of monoclonal antibodies as kind of a stopgap. I mean, they're incredibly important, and the price to make them is coming down, and the way to make them is becoming more streamlined. Some of the other things that are exciting in the monoclonal antibody space is that we're not just putting in regular vanilla antibodies. We are learning how to put in supercharged antibodies that have additional properties and can bring in other components of the immune system that could then trigger somebody to being at a stage where whatever their disease was, I'm not talking just about HIV here, I'm talking generally, is, shifts to a different level and is muted in some way. We saw it in COVID. Monoclonal antibodies were the only, treatments we had early on that protected people who got infected and protected people who were vulnerable. We have, I think it's called EvuShield, which wasn't a monoclonal antibody, but that was a way to provide some protection against at least the early ones. So monoclonal antibodies, are incredibly important and powerful. At the moment they're not. They're kind of, if you like, in competition with antiretroviral therapy. A pill a day. So. But, for some people, maybe an infusion once every 3 months is better than taking a pill a day. We know people have different needs and requirements about in what form they want their medicine. So I'm not going to get into pricing or what, anything like that. But hopefully you've set me up to convince everybody that even though what I'm proposing, which is turning B cells in the body into a factory to make your own monoclonal antibodies, is kind of, even more extreme and potentially at the moment, because it's a gene therapy, massively more expensive. We have to start somewhere. And we hope we're starting to, at least in proof of principle, show that this is possible. Okay. All right. So that set me up nicely to talk about what we're actually doing, which is the idea of, can we reprogram somebody's B cells to make custom and highly effective BNAbs of our choosing? You know, forget what nature wants to make. Let's put in something that we think will be a more potent antibody. Okay, so I want to start off by just saying that, you know, antibodies are naturally made from B cells, and B cells are really clever factories, and the antibodies that make them there's lots of advantages as a result. So let me go through this first of all. Here's my B cell in the middle, making it anti-HIV antibodies. So the first concept I want to talk about is over here. It's the fact that B cells are boostable. You know, if you get infected with a virus or you get injected with a vaccine, what's actually happening there is the very rare B cells in the body that could make an antibody that recognizes that virus or that vaccine component are kind of stimulated by meeting something that they are a good match for. And so the B cells that fit start to grow and divide and expand and become really excited. And so we say they're boostable. So in theory, you know, just one reprogrammed B cell put back into somebody, if it's got the right machinery, will respond to HIV or a vaccine and just spread and grow. So that's really key, because as a gene therapist, it means I can probably be incredibly inefficient and use nature to boost my effect. The second thing I want to tell you about is over here on the right, and this is the idea that B cells, once they have met their virus, or we call it an antigen, the thing that their antibody recognizes, they change, and they change into things that are really great factories. They're called plasma cells, and they just churn out antibodies for many years. So the B cell itself, because this is what its day job is really, really good at making antibodies. And I say that because an alternative approach would be to use gene therapy to put an antibody into, for example, a muscle cell, and have people's muscle cells churn out antibodies, and they could do that as well, but it's not quite as good as having the authentic plasma cell do this. Next thing I want to talk about is down here, this concept of trafficking, which is that B cells, sure, they're whizzing around in your blood, but they also go to places where they would be needed, such as lymph nodes, where HIV could be hiding. So I think having this kind of like Trojan horse aspect of B cells is important as well. And then finally, and this is something I'm not going to talk about today, but I'm putting it in for people who maybe know a bit more about B cells and antibodies. Antibodies kind of continue to evolve. You know, you can put in one antibody and it might get tweaked a little bit and changed a little bit in its B cell as part of its normal way of generating new antibodies. And if it makes an antibody that's an even better fit for your virus, for example, then that antibody will become the dominant one. So over time, antibodies can be enhanced, but I'm not going to talk about that today. Okay, so all these magical properties about B cells and why they are the best cell in the body to make an antibody. The trick is that only happens if our custom antibody is going to be expressed from the same place that natural antibodies are expressed. And that means being expressed from the part of our DNA that encodes antibody genes. And antibody genes are also called immunoglobulins, or the shorthand is IG. So the trick is that we need to take our custom antibody, our custom BNAb that we want the B cell to make, but we need to put it into the B cell DNA at the site of where a natural antibody gene would be. And in the past that was impossible, but today, because we have things like CRISPR, which allows us to do very precise gene editing, this gives us the possibility of doing this. Okay, so I have one little slide as a background on CRISPR, and then we'll get into the nitty-gritty of what we're doing. So CRISPR, or CRISPR-Cas9, to give it its full title, is bringing new capabilities to gene therapy because we can start to put new genes into cells with utmost precision. This is a cartoon of CRISPR-Cas9. Probably don't need to know any of that. The take-home message is that we can program CRISPR-Cas9 to target any specific gene in our DNA. And what it does is it makes a DNA cut at that site. So, for example, we could target the immunoglobulin or Ig gene. And once that DNA break has been made, so now I'm going to go over here, what happens is the cell's DNA repair machinery will fix that DNA break. But at the same time, we can hijack that. We can put in a piece of DNA ourselves that codes for example, our broadly neutralizing antibody. And as part of the repair, it's like we fool the repair pathway, and we end up with a bNAb being inserted in the immunoglobulin gene. So this is the trick that CRISPR-Cas9 allows us to do. Okay, now, there are still challenges when we're thinking about using CRISPR to engineer antibodies at the immunoglobulin locus. And the biggest one is just how damn complicated antibodies are. Okay, so here's me sort of zooming in on a picture of an antibody. And you can sort of see that it's got kind of like 4 chains or strings, 2 long ones that are called heavy chains and 2 shorter ones that are called light chains. So an antibody comprises 2 components, heavy and light chains, and it has to be assembled into this complicated 4-chain structure. So that just makes thinking about doing this engineering so much more complicated. So in order to get around that, the idea that my lab had is we took inspiration from camels, and I guess not many people do this on a daily basis. So why camels? Okay, well, that's because camels and also llamas make a special type of antibody. These are called heavy chain only antibodies, and I abbreviate them as HCAPs for heavy chain antibodies. So look, coming down here again, here's my typical antibody with its heavy and light chains. But now here's my camel heavy chain only antibody. And you'll see it only has a heavy chain. And if you're looking closely, you'll also see that even this heavy chain is kind of a bit shorter. You see, compared to the natural antibody, it's missing this part here. And this part that it's missing is the part that normally interacts and partners with the light chain. So by not having this component, the heavy chain antibodies from the camels they just ignore light chains. They don't need them. They don't interact with them. So much simpler and easier to make, because it gave us the idea that we could, instead of having to mutate and edit heavy chains and light chains in human B cells, we could just focus on the heavy chain region alone. Okay, so the next few slides I apologize for. This is where I as simple as I make it, it's still hard. So hopefully you can bear with me for a couple of slides. So this is showing how we actually engineer the immunoglobulin gene using CRISPR-Cas9. So first of all, this is a very simplified cartoon of what the heavy chain parts of human antibody genes look like. It's got a promoter, which is the front of a gene that drives expression of the gene. And these are the different parts of the heavy chain. I've given them their formal names, but you can ignore those. Basically, what is produced is this heavy chain, 1 red, 3 blue components. And when this then pairs with its light chain partner, this is how we build a conventional antibody. So what we do to engineer this is we come in with our CRISPR-Cas9 machinery, and we are going to insert at a point between the first and second blue components, we're going to put in a new promoter because we want to start making the antibody at this stage here. And we also put in a new head of the antibody, which is our anti-HIV component. So after we've done this engineering, what we end up with is something that looks like this. So you'll see here's the insert, the promoter and the anti-HIV head, and it's now linked to the remaining parts of the heavy chain. And so this promoter now works, and what we get is basically 1 orange and 2 blues, which is basically the structure we have that is part of these heavy chain only antibodies. So that's the gene editing trick we proposed to do. And of course, we had to ask, would it work? So, does it work? So in order to test that, the first thing we decided was that we were going to put an anti-HIV bNAb head, the orange square, if you like, into the immunoglobulin gene. We chose one called J3. I just give you that information because I often call it J3 by mistake, and people are like, what's J3? It's the orange head. And then we wanted to ask if those engineered B cells could indeed make HCABs that can work and neutralize HIV. So the answer is going to be yes. Our J3 engineered B cells do indeed make functional anti-HIV heavy chain only antibodies. I'm just going to give you two pieces of data. When a B cell is making its antibody, it also expresses some of that antibody on the cell surface, so you can detect that on the cell surface. So here we have this is called flow cytometry or FACS analysis, and in cells that have not been edited versus cells that have been engineered, you'll see a difference, and the difference is that these cells can now capture and be stained by, for example, GP120, the HIV envelope protein. So what this is telling us is that these B cells will recognize and stick to GP120. The other piece of data I want to show you is when we take the antibodies that are made by these engineered B cells and put them into what's called an HIV neutralization assay. And the idea here is that the antibodies, if they recognize and block HIV, they will stop HIV infecting our test cells. So what we have here is we are measuring the amount of HIV infection. The black line is when we add what's been secreted by just control normal human B cells that have not been engineered. And you'll see that the amount of HIV infection is not at all bothered by the antibodies that our normal B cells are making. But when we add the antibodies from edited B cells and we increase the amount, you'll see that we get a corresponding decrease in HIV infection, and that's proof that these antibodies are neutralizing HIV. So it works in the lab. So the last thing I want to sort of let's recap before I tell you the last part of my talk. So we've copied the design of the heavy chain only antibodies found in camels to allow us to customize human antibody genes in human B cells. And we've shown that those engineered B cells at least have the potential in the lab to secrete anti-HIV antibodies and specifically anti-HIV bNAbs.
Paula, before you go on, we had a question. People were asking, is this editing that you're talking about that you've proven, was that done ex vivo?
Yes. Oh my gosh, that's a good question. It was done ex vivo, but we are also trying to develop a way to do it in vivo because, even though, do you remember I said that because B cells can be boosted, in theory, this is what I love about them. You could probably go in with, in theory, but, a very small number of B cells. So even when I'm thinking about doing this ex vivo, I can imagine a scenario where, you're taking a small amount of blood from somebody, pulling out the B cells, editing them, and then popping those few B cells back in. But even better would be if we could inject the CRISPR-Cas9 B cell editing machinery into somebody and have it just go to, a few random B cells it bumps into and engineer those B cells in vivo. That would be so much simpler, and that's ultimately, what we wish to do. But baby steps.
Yes.
So our next baby step is we've shown this in the lab is thinking about how to show this in an animal model. And those of you who've heard me talk before are probably expecting that what you're going to see now are some humanized mouse data. Because the lab works with humanized mice a lot, but unfortunately, human B cells don't work well in humanized mice. Humanized mice have many of the components of a human immune system, but they don't have all of them. And one of the things that doesn't grow well in humanized mice are functional human B cells. So unfortunately, we're gonna skip humanized mice, and instead the only animal model that's really going to be able to allow us to answer these questions is going to be the rhesus macaque. So what are we doing next? Well, with funding from both of the collaboratories I'm part of, we are developing our pilot studies in rhesus macaques. So here's my last slide, I hope, showing how we're adapting this strategy. So basically, here's a rhesus macaque. We're going to take some B cells from its blood. Ex vivo in the lab, we do our gene editing with our custom heavy chain only antibody. Put those B cells back into the macaque, and then the sorts of studies we'll do is we will vaccinate the macaque to see if those B cells respond to vaccination. And then we will also challenge the macaque with, in this case, it would be SHIV, but we could also do it with an SIV. So the questions we have are, first of all, the first question we had was, can we figure out how to engineer rhesus macaque B cells? Because we've been doing human cells so far, and the answer to that is yes. We spent the last 18 months adapting our technology to macaque cells. Can those engineered macaque cells survive when they're put back into the macaques, and do they respond, do they get boosted if we vaccinate with HIV GP120? Do those edited cells after vaccination secrete enough of these HCAB antibodies that we can detect in their blood that they could then protect against a challenge with SHIV? And then ultimately, can the edited B cells, if we then give them to an animal that's already infected, can they help to control that infection in an already infected animal, which is what we'd like to do. Okay, so just some final thoughts on what we hope. Okay, I've said this several times already, but I hope that even very low levels of engineered B cells will be enough, and that they'll behave like regular B cells and become boosted in response to either the presence of HIV or a vaccine. I hope that the engineered B cells in the body will turn into these plasma cells. Do you remember these are these long-lasting factories that will produce antibodies for many years? I also hope that we can give mixtures of B cells, making a blue antibody, a red antibody, a purple antibody, so we can put in a combination B cell therapy to give us better coverage and present a bigger challenge to the virus, hopefully an unsurmountable challenge for it to evolve resistance to these different antibodies. And then beyond HIV, we're thinking about this platform for cancer and any other diseases that are currently treated by injections of monoclonal antibodies. I gave you one example, inflammatory bowel disease, and also for certain immune suppressed individuals who can't make a needed antibody. And we've seen this especially with, for example, COVID antibodies. So that's my long wish list for this type of therapy. So I'm going to end on 2 slides. First of all, just thanking the members of my lab that are funded by RID-HIV, which is the main collaboratory, because I'm one of the PIs there, but also recognizing I have amazing colleagues in I4C who are helping me to engineer the best BNAbs to put into our B cells. And finally, I'll end and leave up the summary for the community. Hopefully I did what I said. Again, our question is, can we reprogram B cells? Our answer, yes, we can, at least in the lab. Why is it important? Lifelong production of antibodies to block HIV. As a potential cure therapy, it's because antibodies can neutralize HIV. And if we put in really good custom antibodies, we have a way to improve on nature. And why should we be excited? Because again, I think it's a completely new idea. I love this idea that we can improve on nature. We've not engineered B cells before, and it could also work for cancer and other diseases. Okay, and with that, I will stop sharing so I can see all your lovely faces, but I'll pull up slides again if people have specific questions.
That was really wonderful, Paula. I mean, I think it was one of the best presentations we've had as far as people understanding what it is that you're talking about. And actually, I thought it was a really good idea to put that slide not only up front, but to reiterate everything at the end. So I'm going to remember that. Thank you so much. Let me ask you just one question, and then we'll get to the questions in the chat. Maybe people can can ask them if we have, or, you know, Michael, maybe you might want to read them since we.
Yeah.
I've got like 57 things in my chat, so I can pull out.
Yeah, but is there any danger of this increased B cell function that you know, I mean, I think that might be the problem of what's going on with HIV, overreaction of some of this stuff, of all these immune system functions until it flops out.
That's actually a really good point, Linda. So, you know, I'm a cheerleader for this technology, but it's very early days. And whenever you do something to the immune system, you always have to worry about unforeseen effects, including, if you like, hyperstimulation. I think and that's of course one of the reasons why we would do a lot of safety studies in rhesus macaques. What I can say is, you know, I've alluded to this a couple of times in my talk, is that, you know, we are not limited to putting in natural antibodies. We can put in antibodies that have both enhanced function, but also we can put in antibodies that have maybe some built-in safety features. You know, antibodies do more than I've told you. Okay, full disclosure. I've talked about, you know, this really important activity of just binding to and blocking HIV, neutralizing HIV. But antibodies do other things, including, for example, you know, when they will also recognize HIV on envelope protein, GP120, on the surface of an infected cell, and they will stick to that. And, you know, antibodies are these little Ys. So the tail of the Y is, if you like, sticking out. And other parts of the immune system recognize if a cell is coated with antibodies because the cell is expressing something foreign. Other parts of the immune system will come and take a look at that as well, including things like macrophages and natural killer cells. So these other properties of antibodies as a way to kind of corral and bring in other components of the immune system are important. But one of the things you can do, we could play it really safe, and we could engineer antibodies to not do these other things. We could say, you know, that might just be too much. It might be we create an immune storm that might be too much. So, you know, early on we have options to engineer our antibodies to also not do these other things and start, if you like, really slow and safely. But you bring up a really important point, Linda, and I remain humbled by what we don't know about the immune system. And so when we start to go in and engineer it, absolutely, safety is our first consideration.
Thanks. Michael, you want to go ahead and...
Sure. So I'm going to go back to about 9:45. We had a question. How effective are these engineered antibodies compared to naturally occurring antibodies?
Okay, that's a great question. So because, you know, there's two things. I've told you that we're making these like mini antibodies, these heavy chain only antibodies. So the good news is that people have been messing around with camels and llamas for a long time, including the idea that they could vaccinate llamas and make some of these natural heavy chain only antibodies as a way to have a new class of injected monoclonal antibodies. Just because, you know, they're simpler, they're much easier to make than regular antibodies, even when you're making them as a monoclonal antibody to be injected. So there's already been a multitude of very potent, broadly neutralizing llama and camel antibodies made. And the one I selected, J3, we didn't choose that by random. We got a whole panel of natural camel antibodies, and we tested them ourselves in the lab. And J3 was by far the best antibody that we found, both in terms of its potency, but also its breadth. You know, it neutralized lots of different variants of HIV, so it really deserved the title of being a broadly neutralizing antibody. And then last year, the reason for that became clear because another group had done some studies asking which part of the HIV GP120 does this J3 heavy chain antibody actually bind? And guess what? It binds to that. Do you remember the CD4 binding site, the yellow oval I showed that I said is that's a bullseye for antibodies because the virus would really struggle to mutate that. Well, it turns out that J3 is a broadly neutralizing CD4 binding site antibody. So we chose wisely, I guess. So there's ways to make even more effective ones. But heavy chain only antibodies, they're also called nanobodies, are increasingly being developed and used in the clinic for other diseases. And we just keep an eye on all the improvements, and our platform can easily swap out and swap in the best antibody that's available. So I think it's going to give us something that's potent and broad.
Great. Another question is, are these heavy chain only antibodies not immunogenic themselves? And what about anti-idiotypic antibodies?
That's a great question. So one of the problems with antibodies is, in theory, if you put antibodies, especially from a different species, into somebody, the antibody itself can be recognized by antibodies. It's kind of crazy, isn't it? So, and that's what are called anti-idiotypic antibodies. So there are ways around that. One is that we talk about humanizing antibodies. You may have an antibody that was originally developed in a mouse or a llama. But what we can do is, once you've got that antibody, we can make subtle changes to it that make it look like a human antibody, and therefore it's much less likely to produce antibodies. So it's theoretically possible, of course, and we would, if and when we go into humans, I guess the first thing I would do is I would propose injecting, for example, just the humanized J3 antibody, for example, alone into people. Forget edited B cells, let's just put the product in, and asking whether or not that was well tolerated, whether it suppressed HIV, and whether people didn't make antibodies against it. I'm sure, again, as you guys know, that we've already done clinical trials with conventional bNAbs, and I think there will be clinical trials with heavy chain only antibodies as a next generation of bNAbs. So we will make sure that when we put in engineered B cells, we are using our custom antibody payload is something that's already gone through that step of development and been shown not to have that problem.
So another question that came up, removing the light change can reduce the human antibody potency. Do you know how well these heavy chain antibodies work compared to the full chain antibodies? And I think
so that's a little bit like the previous question I answered, which is just saying that, there are some heavy chain only antibodies that are incredibly potent, and we just make sure they're the ones we select.
And someone wants to know about the persistence of engineered B cells once injected into the body. Do you have any clues as to how long they may be lasting? Any adverse events?
This is the sort of information we'll get from the rhesus macaque studies, because we'll be asking, do the engineered B cells persist and thrive? Remember, we want them to persist. We also want them to be boosted and respond to vaccination or HIV or HIV infection. So we can obviously, we can use genetic tags to measure how many of the B cells are our engineered B cells, and we can follow them over months, if not years, in a macaque model to answer those questions. That will all be asked in rhesus macaques before we go into humans.
And someone wanted to know, if this treatment fails in the macaque model, does that mean that it will fail in humans and will therefore be abandoned?
No, I mean, I expect it to fail. I mean, I'm a scientist. Everything I do fails, but you fail wisely. If you do an experiment well and you collect enough samples and you've already thought about what could go wrong, when it goes wrong, and it will, we should have the data that can allow us to then go back and tweak what we are doing and change some of the things that are the reasons for the failure. So I anticipate failure, but I anticipate failure with information that will help us to get to the next stage.
Another question. So for optimal performance, the B cells need also T cell stimulation. Have you considered applying peptides to provide optimal T cell stimulation?
Oh my gosh, that's such a sophisticated question. Absolutely. We have another project in the lab where we are engineering our B cells to be, if you like, T cell independent, so that they are like little self-contained super B cell. And we're doing that first in the cancer space, because we are creating a Frankenstein B cell. Oh, I shouldn't call it that, you'll quote me, but because we are creating a B cell that has enhanced, never before seen properties, we would only do that in very serious and severe cancers initially. But yes, that's a great question.
Yes. There was another one. Where did it go? Do you expect that these engineered B cells and plasma cells will be able to cross the blood-brain barrier?
Oh, we also have a project doing that. So yes and no. One of the things our approach is that there are certain, remember I keep saying we can fully customize these antibodies. You'll have to invite me back and I can talk about that project. So we can make something that really are non-natural antibodies but have enhanced abilities to cross the blood-brain barrier. So a new project in my lab is absolutely looking at that. We are engineering our B cells to make antibodies that can cross the blood-brain barrier. And that one we are testing in humanized mice because we have a nice humanized mouse model. We work with Jonathan Kahn's group on that, which has HIV infection in microglia, which are the cells in the brain, so we have a good readout. So these are great questions.
Yeah, and we had this question was asked twice. People wanted to know about reinfection, how reinfection would affect these treatments.
How reinfection would infect the treatments. So you mean if somebody was infected with HIV and then got exposed to another round of HIV, do you think?
I'm assuming that that's what they mean.
Right. So hopefully, in the same way that, it's actually, I'm not aware, I imagine it's incredibly difficult to get reinfected because if you're infected with HIV, you are making antibodies and immune cells that would attack any new HIV coming in. So because we are trying to make B cells that make even better antibodies that are broadly active against multiple different strains of HIV, that should stop a reinfection, but I don't know how common reinfections are anyway. But the idea is that, well, gosh, this is both a prevention and a treatment. Remember the studies we're going to do in the macaques? The first thing we're going to ask is whether or not our engineered B cells and the antibodies that they are producing can actually work as a sort of a vaccine, can prevent people from being infected. That seems like a very extreme way to protect people. Put a condom on is always easier, but we would show that because that's an easier thing to demonstrate in an animal model. And then we would look at whether or not we've made an immune therapy that works for an already existing infection.
Well, we have many more questions, but we have reached our time limit, so I'll have to cut them off here. Maybe I can send them to you via email and you can respond that way.
People can email me.
Yeah, great.
Directly as well. I'm pcannon@usc.edu, or you can Google me. You'll find my email. If I don't reply straight away, it's because my inbox is full of hate mail from COVID anti-vaxxers at the moment. And so I get something like 200 emails a day. It's quite a challenge to go through.
No.
It's all right. Well, how can I say? I'm a woman who likes vaccines. I'm very triggering to some people. So maybe just put not a COVID.
If people either love you or hate you, that's fabulous.
Well, yeah.
Good.
Thank you, Linda. Well, hopefully, and thank you for these great questions and your attention. And
thank you so much. It's wonderful. It's been really, really wonderful. Great questions too, everybody.
Yes, really wonderful. Thank you so much for such a great presentation.
And our last webinar. Oh God!
Well, we might have the extra one from American Gene Technologies.
Well, I think we're gonna. Aren't we gonna do that as a cab call? I think.
Okay.
Yeah, I think. I thought we talked. I think I talked to Michael about that.
Yeah, you
but
did.
We can. That's. For now, the last scheduled one is gene therapy in Africa with Boro Dropluck from Caring Cross, who a number of us are involved with that organization. So, in other words, we had talked a lot about pricing today, and it's about how to bring, as far as the scalable thing, how to be able to get these therapies to developing countries. So it's really very, very interesting. It's a whole new way of pricing these things. So anyhow, or doing things that that make that pricing so much less costly. So that's February 28th, and we hope you will all join us. Thank you so much again.
Yes. And we will be putting this video up on to Facebook and on YouTube, so you'll be able to access those. I'll be sending out an email that'll also include the webinar evaluation form. If you didn't catch it in the chat there, you'll get it through the email. And you can look for that email later today. Hopefully, I'll get this all done by the afternoon. So thank you for joining us, everyone. Hope you had an edifying experience. I thought this was really lovely.
Great.
Good to see you
Thanks,
all.
Michael.
Thank you all.
Thank
care.
you all for coming. Michael, can I call
Thanks.
you?
Bye-bye.
Yes, you can.
Okay, all righty. Bye, have a
All right,
great day.
take care, everyone.